Okay, thank you, Mark, for the introductions. Good afternoon, everyone. I'm Yaomeng Liu. I'm a PhD student from Oxford Brookes University, and today I'm going to talk to everybody a bit about exosomes in Parkinson's disease. Okay, let me start from introduce what Parkinson's disease is. I think everybody here must have a knowledge about this more than me even. Okay, so Parkinson is the second most common neurodegenerative disease, just after Alzheimer's. And in every 500 people in UK are affected by the Parkinson's disease. People with Parkinson's disease, they will have a higher risk to have the dementias in their later lives. And also like the Alzheimer's disease, Parkinson's also have the mechanisms including the reactive oxygen species, have the neuroinflammations, and also the apoptosis. Yeah, and we all know that exercise is very important in our lives, our daily lives. The regular exercise can help a lot to help us to prevent the chronic disease. This could be because our muscles, our skeletal muscles, can be worked like secretory organs. After exercise, those muscles are going to secrete or produce some cytokines, some peptides, and release those small vesicles into the circulations to help us to build our bodies. And the transport of these cytokines may be done by the exosomes. So here let's have a look at what exosomes is. They are very small, tiny vesicles in our bodies. These can be reduced by a variety kind of cells. We can see from these pictures, this is how a cells produce the exosomes. Yeah, we can also have a look at the outer layers of the vesicles to find out where they come from, what kind of these vesicles is, all the information. Also, they are including varieties of proteins and RNAs inside as a component inside those vesicles. So from analyzing the components of those vesicles, we can find out what information they carry. Like I mentioned before, after exercise, your muscle may release some of the information, proteins, and transport them to the circulations. And this could be done by the vesicles because those small vesicles will just act like a cargo, form those informations and go to the circulations to help the cell to do the cell-to-cell communications. And this picture shows the exosomes and the Parkinson's disease. So the exosomes can be isolated from different kind of cells and also body fluid, like the plasmas, like the urine, salivas. And by looking at the component of those exosomes from body fluid and aimed at a different target, we can distinguish Parkinson patient and the normal control patient. So it's like a very novel biomarkers to help us diagnose the Parkinson's disease. And these are the papers that published in recent years about the biomarkers of exosomes. So in our groups, we used to do the studies to have a look at the effect of exercise on Parkinson patients. This paper was published in 2017, and on this paper, the rescreening a large numbers of people with Parkinson's and also age-matched controls in Oxfordshire, we screening them, and then we exercise them 3 times per week, up to 12 months in total. And we find out differences between the normal age people and the Parkinson patient. We found that the Parkinson patients have got the blunted exercise capacities, and they have to reduce their cardiovascular and metabolic response. Also, they reflected by lower oxygen utilizations and heart rate, even we push them to exercise to the max of their levels. And this work has already been published, so the next step, the next study of our group, our teams, is going to have a look at the differences in cell levels and also the molecular levels. So here is the new experiment designs. We're still going to recruiting the Parkinson patient, including the early onset or the late onset Parkinson patient, and we are going to recruit in the healthy controls, age-matched healthy controls as well, and we invite them to come to our lab, set them on the bike, wear the masks, and we are going to molecule the heart rate, the oxygen utilities, and also the carbon dioxide produced during exercise. Also, at the same time, the lactate level, the glucose levels during the exercise will be measured by every 2 minutes. Also, we are going to take some of the blood samples and isolate the plasmas, and then isolate the exosomes from those plasmas before and after exercise. Have a look at those exercises. Are there any differences between those exercises of healthy controls and the Parkinson's? What's the component before and after the exercise could induce? And because it's a long testing, it takes a long time, and we know the lab is a very different environment for the patient, also for the participant. So I even bring my teddy bears there to try to relax them and make them feel more relaxed about the environment. And currently, I need to make this as examples about 2 of the special participants we have. They are aged and gender matched controls, and they are quite physically, they are quite exercise. So here is a short videos about them. They used to rowing across the Indian Oceans before they come to our studies. Here is a quick videos from the BBC's.
Life doesn't stop. You can still do things, you can still live your life and have a full and meaningful life as well. I honestly thought I'd forgotten how to walk. I started limping and dragging my foot.
At the time we said, right, we're going to do this, 10 times the amount of people have been in space and across the Indian Ocean.
Most people would think, wow, that's impressive just for an able-bodied person. Wow! The best bit was when 2 whales actually came over, and they were inquisitive enough to come over and see what we were about.
That is insane, look at that.
He's walking pretty much normally.
It has made me better in the short term, and I just feel fantastic for doing it.
When you're ready, just walk as you normally would.
It's clear to say that exercise is just, for me, it's the wonder drug.
Okay, at the very last seconds, we can have a look at how we run these studies in our lab. And we invited them to our lab and have the testings, got some samples, and have a look at that. So both of these 2 participants, they are very they are very activities, and they all finished the testing, the exercise testing at the same levels by 22 minutes. But what we can look from the result got from this study is that we monitor their lactate and glucose levels every 2 minutes. We also monitor that after they finish the exercise testing, we can find out that the Parkinson patient, they got the slowest recoveries after the exercise, like his lactate levels still keep very high after exercise, while the healthy controls got that back to normals very quickly. And this result matched our previous studies about they are going to have a slow recoveries. And the next step, future work in our lab is like we are still recruiting Parkinson patients, and we are also trying to push these studies, trying to get the result from the exosomes from their plasmas and have a look at are there any differences between before and after exercise and between patient and health controls. Okay, thank you very much for everybody here, and thank you for my supervisor team sitting in the middle of you. Okay, thank you.